dextrin, chitin, and agar. Like the agar we're using in the lab, that is also example of a polysaccharides. Okay, so those examples we'll talk about detail later. Okay, here is something I want to skip it because we don't want to see. I cannot see it. Nucleic acids. The slides I'll just skip it because we already know DNA and RNA. Okay, ATGC. You know that A complementary with G. A complementary with T, C complementary with G. They are composed by hydrogen bonds. And let's talk a little bit easier. DNA, double helix. Okay, A complementary with T, G complementary with C. This is a hydrogen bonds. And because of the GC has a 3 hydrogen bonds, so when we design the PCR primer, we usually think about the components, the content of GC. Okay, that's hydrogen bonds. And then the double helix, which is anti-parallel, always is one coming from 5' to 3', another one 3' to 5'. Okay? And the rounding opposite direction and the composition is actually referring to the 5-carbon sugar, the deoxynucleotides. And how they do replication is semi-conservative. That's what we know. Semi-conservative. Okay, we easily talk about DNA. RNA most of the time is single bands. They do not have G. They do not have T. They have uracil. Okay. Now, when we have a vaccine right now for COVID-19, the majority of the stuff, which is mRNA, it's a little bit different from the vaccine what we had before, because the vaccine mRNA come into the body have to be translated, become protein. The S protein will be targeted for the COVID-19 virus. Is that right? But there's lots of reports that they have a very strong side effects because it's relatively new. Because the mRNA going into the body have to be translated. That's everything could happen. Remember, we have we call it lactose operant. That's in bacteria in the human body is more complicated than that. So that's brand new stuff. Okay, so that just want to mention. Okay, last part we want to talk about is lipids. Then we will have a special section talk about lipids. But I just want to talk about here. Now, lipids, when we talk about lipids, this is a nonpolar. And this is hydrophobic. So they dislike water. You understand it's not going to dissolve into water. It's dissolved into organic chemical solution, like ethanol typically, or benzo, or chloroform, those stuff. Okay? What are they looks like? I'll give you a very simple example. What is this? Glycerol. Okay, so what are we going to have? Oh. What is this? Fatty acids. Okay, when you combine, what we do? This water is gone. Okay, so dehydration. then becomes what? That is all. Actually, it's all 1 or 2 or 3. It may be different, it may be the same. This is the one we call it triglycerides. Okay, that's what happened. This is going to take out the 3 water. Okay, that's triglycerides. So glycerol with 3 molecular fatty acids, that could be the same, could be different, become triglycerides. That's the overall reaction. Now, what we're going to talk, just give you a very simple conclusion. Lipids could be solid, or we say saturated. That's a good example is fat. Could be unsaturated. A good example is oil. Is that right? Okay. What means saturated? No double bonds. What means unsaturated? Have double bonds. Okay, that's very simple, very simple. What a good example, palmitic acids, this is oleic acids. We just say fat and oil. Now, unsaturated fatty acids can be saying, if the double bonds is only 1, we call it mono unsaturated fatty acids. If the double bonds is more than 1, we call it poly unsaturated fatty acids. Now we have another topic. Okay, we have a saturated fatty acids, usually like this. If they like that, the 2 hydrogen bonds is on the same side. This is called a cis. If this happens, that's called a trans. Okay, trans fatty acids is not good. That's 1988 introduced into the bread manufacturing. The idea will be prevent the fatty acids oxidation. That's called the resinidate process. So kind of like a chemical stabilization products to extend shelf life of bread. But trans fatty acids is not good because the liver do not have the enzyme can break down it. Finally will become the so-called blood cell clotting, will cause all kinds of cardiovascular disease. Is that right? So trans fatty acids is not good. We will like the system. Okay? But there are some of the good things happen. Usually we say unsaturated fatty acids is good because it's easy to be hydrolyzed and digested in the body. Saturated fatty acids is not good. But trans is an exception. Okay? So that's what we talk about. Okay, phospholipids. This is something you know that. So let's say we had a bacterial cell. This is cell membrane system. Now I talk about this all the time. So this is protein. Here we have some, what is called? Lipopolysaccharides. Oh, sorry, that's called the phospholipids. These guys we call phospholipids. And these phospholipids, you have this head and the tail. This head is hydrophilic, which means they like water, and that tail is hydrophobic. phobic, and they dislike water. And that protein is inserted in there, could be a supporter, but more important is for the chemicals go inside of the cell membrane. And this is called fluid mosaic model. We know that, okay? So that's phospholipids. Now, lipids also contribute for bacterial cell, including lipoprotein and LPS, lipopolysaccharides. That is why the salmonella is a big trouble, because the lipopolysaccharides contribute to endotoxin. Okay, that's why LPS is a big trouble. That's a lipids combined with protein, actually. Sorry, the lipids combined with polysaccharides. The lipid combined with sugar. Now the last one, I don't have anything, I'm not going to draw. We will talk a little bit about steroids. They have a certain ring, it's a stereo ring, it's like this, you can draw. We have this ring. Do like this. Okay? This is we call steroid ring. And this steroid ring, if they are adding an OH, which is a hydroxy base there, this has become sterol. What's a good example for sterol? Cholesterol. Cholesterol. Is that right? We don't like that. Do you know LDL, HDL? Those not good. So LDL you have to be keep lower. If more than 60, probably you have to do some diet control. Otherwise, you will have a cardiovascular disease. HDL is good. You have to keep it higher. Sorry, LDL should be less than 200. HDL have to be more than 50 to 60 because they're going to remove all the clots, and we call it a good fatty acids, and LDL is bad fatty acids. That's cholesterol. That's not good. Then other steroids. A good example, cortisol. So we say it's a cortisol. What is cortisol? You can buy one cortisol from CVS or Walmart or any of the drugstore. If you have an itching of your skin surface, you add 1% of those products. But be careful, the cortisol is a hormone. Any of the products which can protect the skin has a hormone there. So that's why they keep 1% for adults, 0.1% for infants. Okay? That's cortisol. Progesterone, another example, is steroid. That's, you know that. Progesterone. Progesterone, you know that. If a woman who has repeated miscarriage, and usually the doctor could provide progesterone to prevent that, possible, if the reason is lower level of the progesterone. Okay? That's an example of the steroids. Okay, so that's all I have today. So,